Jeffrey Smith, Executive Director of the Institute for Responsible Technology, spoke to over 800 healthcare professionals at the 2011 Nutrition and Health Conference, sponsored by Andrew Weil's Arizona Center for Integrative Medicine. His topic was the documented health risks of genetically modified foods. Mr. Smith is also the international best-selling author of Seeds of Deception and Genetic Roulette.
So what we're going to describe today is a summary of some of the health risks, an overview of the things that can go wrong, and we'll reserve the more detailed descriptions for articles, etc. There's basically has been 4 major genetically modified crops: soy, corn, cottonseed, and canola. And there's also some minor crops: Hawaiian papaya, a little zucchini and crookneck squash, and more recently, sugar beets. 95% of the sugar beets in the United States are genetically engineered. Now, I'd like you to rate yourself personally from a scale of 1 to 100, how vigilant you have been at avoiding genetically modified foods. Now, on a scale of 1 to 20, most Americans are -6. And most Americans don't realize they've ever eaten a genetically modified food in their lives. So I'm going to ask you to raise your hand if you're low vigilance. That should be most of you, 1 to 20. Please raise your hand if you're low vigilance. Thank you. Okay, 20 to 40. 40 to 60, 60 to 80, 80 to 100. All right, that's a pre-test. We'll ask the post-test in about 40 minutes. All right, I'd like you to rate yourself how active you've been at educating your patients on the risks of GMOs. That also should be low, 1 to 20. How many people are 1 to 20? Raise your hand. 20 to 40, 40 to 60, 60 to 80, 80 to 100. All right, so I've got some work to do. The reason why GMOs are on the market is because of a single sentence in the FDA policy that came out in 1992, which is still in force. And it was a lie. It was a complete fiction. It said that the agency is not aware of any information showing that the foods created from these new methods differ from other foods in any meaningful or uniform way. On the basis of this sentence, the FDA policy says no safety testing is necessary, no labeling is necessary. In fact, if you create a GMO in your basement, you can put it on the food supply without even telling the FDA. This is their process of regulation. 44,000 secret internal memos were made public from FDA files 7 years later, showing that in reality, the scientists had warned over and over again of potential dangers of GMOs, allergens, toxins, new diseases, and nutritional problems. They had urged their superiors to acquire a long-term study and complained vehemently about the policy. So where did the policy come from? The White House had instructed the FDA to promote biotechnology, so they created a new position for Michael Taylor, Monsanto's former attorney, in charge of FDA policy, then became Monsanto's vice president. Now he's the US food safety czar in the Obama administration. The first genetically modified crop to participate in the voluntary consultation process of the FDA was the Flavor Saver tomato, and it's the only company that gave raw feeding study data to the FDA for a rat feeding study, and it turns out the rats refused to eat the tomatoes. And it's not just rats that seem to have an inherent wisdom about GMOs. Eyewitness reports from all over the world show that animals like pigs, cows, geese, squirrels, elk, deer, raccoons, mice, rats, and buffalo are all given, when given a choice, avoid the GMOs. So it's our job to get humans up to the level of animals. I was, in my book, Seeds of Deception, before every single chapter, I have a little vignette, Wisdom of the Animals, where I describe another example of this. And someone tried to repeat the experiment with squirrels. And they put bags of GM corn on the cob and non-GM corn on the cob in their workroom waiting for the winter. They forgot about it. And when they discovered the bags, the mice had done the experiment for them, breaking in to the non-GM corn and eating it all and leaving the GM corn untouched. So they force-fed these rats the tomatoes that had refused to eat the tomatoes. 7 of 20 developed stomach lesions. 7 of 40 died within 2 weeks. Now, in the UK, they decided to give $3 million of grant money to Dr. Arpad Pusztai, the world's leading researcher in lectin proteins. He worked at the Rowett Institute, one of the leading research institutes for nutritional research. And his job was to figure out how to test for the safety of genetically modified foods. And the protocols that he was to create were supposed to be implemented into EU law. And as part of his research, he took a potato engineered to produce an insecticide, which he knew to be harmless. And he took one group of rats and fed the genetically engineered potato with a complete and balanced diet. Another group was fed non-GM potatoes, and a third group non-GM potatoes plus the insecticide that the GM potato produced. So you have GM potato, non-GM potato, and non-GM potato plus insecticide. Only those that ate the GM potato got sick. What was the cause of the problem? It was not the insecticide. It was somehow the process of genetic engineering that caused potentially precancerous cell growth in the digestive tract, smaller livers, brains, and the testicles, partial atrophy of the liver, damaged immune system in 10 days. Here's a picture of the intestinal walls, the proliferative cell growth on the right from the rats that ate the potato. It's more dramatic in the stomach lining. Dr. Pusztai went public with his concerns and was a hero for about 2 days. And then 2 phone calls were allegedly placed from the UK Prime Minister's office, forwarded through the receptionist to the director of his institute. The next day, Dr. Pusztai was fired from his job after 35 years. Silenced with threats of a lawsuit, his team was disbanded. They removed the data. They never implemented the protocols. Instead, they embarked on a campaign designed to destroy his reputation in order to protect the reputation of biotechnology. 7 months later, his gag order was lifted by an order of Parliament. He got his data back, and now this is published in The Lancet. But what it shows is that the process itself is inherently dangerous. I asked Dr. Pusztai, what was your most shocking moment in all of this? Can you guess why I asked that question? Because I was writing a book. And I was looking for shocking moments, like the UC Berkeley professor who said that a senior Mexican official threatened him, implying, "We know where your children go to school," trying to get him not to publish evidence of contamination of the corn in Mexico. And I have 2 Fox TV reporters in Florida who say that we're about to do a 4-part news series to blow the whistle on bovine growth hormone, which has higher levels of pus, higher levels of antibiotics, higher levels of both bovine growth hormone, and higher levels of IGF-1, which is linked to cancer and fraternal twins. But 3 days before they were to air the show, threatening letters from Monsanto's attorney promising dire consequences to Fox and Rupert Murdoch canceled the series. Or the fact that an epidemic in the 1980s around L-tryptophan that killed about 100 Americans and caused 5,000 to 10,000 to fall sick. When the FDA testified before Congress, they failed to even mention that the only brand that had caused the disease was from a genetically modified bacteria. So I was looking for shocking moments, and it turns out that Dr. Pusztai had one. But it wasn't being fired. It wasn't discovering the problems with the rats. A few months earlier, he had been given the opportunity to read the confidential submissions of the biotech industry for their products that had been approved. He said to me, "Jeffrey, that was a turning point in my life. I realized what I was doing and what they were doing was diametrically opposed. I was doing safety studies. They were doing as little as possible to get their foods on the market as quickly as possible." And a few months later, he realized that if his potatoes had been tested by industry, they would have made it to the market. But more importantly, the soy and corn and tomatoes that had already made it to the market had been produced from the same process that he had used to produce the potatoes. And it was not the insecticide, it was somehow the process that caused the problem. Now, this is the first of 5 categories that we'll look at today, the process itself. What is it about the process of genetic engineering that might be unsafe? Well, when you insert a gene through a gene gun or bacterial infection, and then clone that cell into a plant, you create massive collateral damage. Approximately 2 to 4% of the DNA is different, mostly mutations. Genes can be switched off, switched on, damaged, altered. And one study on a human cell found that when they inserted a single gene, up to 5% of the active genes changed their levels of expression. They were shocked about this. It's a rare study. And when you insert a gene into crops, we would expect the same thing, where you can have hundreds or possibly 1,000 genes upregulated or downregulated significantly. Now, this, of course, changes the RNA and the proteins and the natural compounds. So it's a huge variable that's not being accounted for, that's not being looked at before the crops go on the market. For example, in the most popular BT corn, that's a corn that's engineered to produce a toxin that kills insects. It breaks open their stomach and kills them. After it was been on the market, they found 43 proteins that were significantly upregulated or downregulated, including gamazine, which turns out to have been switched on in the GM corn, which is a known allergen. But that only elicited an interestingly. The fact that GM corn has a new allergen was only interesting to the researchers. What was a major concern was that existing proteins had been truncated and their shape had been changed dramatically, which of course can change a harmless protein into a deadly one, and no one followed up to see if that was the case. In soy, they have a practically doubling of soy lectin, which is an anti-nutrient. Trypsin inhibitor was as high as 7 times more in cooked GM soy compared to non-GM soy. Lignin was increased in soy as well as in corn. Now, the metabolic pathway that produces lignin produces rotenone, which is linked to Parkinson's. And no one has checked to see if GMOs will increase risks of Parkinson's. So those were unpredicted side effects no matter what you introduce to the crop. The second cause of problems is very specifically what it is that you're introducing to the crop on purpose. Which protein? Now, there's 2 major traits. The first is herbicide tolerance. Here's the backstory. Monsanto found bacteria growing in a chemical waste dump near their factory in the presence of Roundup, which normally kills bacteria. So they figured, great, let's put it in the food supply. So they took the gene out of the bacterium that allowed it to survive Roundup, put it into soybeans, which are now called Roundup-ready soybeans. So you can spray the field with Roundup and kill all of the other plant biodiversity, but not the soybeans. And most of the genetically modified crops on the market are herbicide tolerant. That is the reason, the major reason, why we have GMOs, to sell herbicide. The other is this Bt. Bacillus thuringiensis is the name of soil bacteria that produces a toxin which kills insects. It's used in normal agriculture by gathering up the bacteria and spores, spraying it onto a plant. It kills the insects, then it washes off and biodegrades. But the biotechnologists take the gene from the Bt and put it into corn and cotton. So let's take a look at why they think it's safe to put an insecticide directly into every cell of corn and cotton. First of all, they point to the fact that even organic gardeners use Bt spray, and it has a history of safe use. They also claim that it's destroyed during digestion in mammals, and there's no receptor cells in humans or mammals, so it's completely safe and benign. Well, it turns out that's not true. When it was sprayed in Washington and Vancouver, about 500 people reported allergic and flu-like symptoms. Some had to go to the hospital. Farmers mixing the Bt had accidents and had to go to the hospital. When it was fed to mice, it not only survived digestion, but caused tissue damage and immune system responses. But the Bt that's in the crops is thousands of times more concentrated than the spray form. It's designed to be more toxic. It even has properties of a known allergen, which the WHO criteria says should not have allowed it to be on the market. Now, when mice eat the Bt corn, as opposed to the non-Bt corn, they too get multiple immune system responses. IgE, IgG, cytokines, T cells, B cells, a whole host of serious problems. Mice eating Bt/Roundup Ready corn had fewer babies and smaller babies. And when you look at Monsanto's own rat study, as was done by a French team, they found signs of toxicity, immune system responses, etc. Now, in India, thousands of farm workers who pick the cotton that's genetically engineered to produce this Bt toxin are reporting allergic and flu-like symptoms, and some have to go to the hospital. We see pictures over and over again of rashes all over the bodies of these workers, on the faces of babies, etc. In fact, when I looked at the doctor's reports, having analyzed 23 Indian farm workers with these symptoms, and I compared them to the 500 people who reported allergic and flu-like symptoms in the Pacific Northwest, the list was nearly identical. When they allow sheep to graze on the cotton plants after harvest, thousands of sheep died. In one small study, an NGO, a non-governmental organization, had pleaded with the government to do a study. Even though the Andhra Pradesh government acknowledged the sheep deaths and put out warnings, they didn't do a study. The NGO offered to pay universities for the cost of doing a study, and no one did it. So they did their own study. They tied up 3 sheep in GM-BT Bolgard 1 field, 3 in Bolgard 2, and 3 in non-GMO. By the end of the month, all the 6 eating the BT plants died. The 3 eating the non-BT had no symptoms. But it was dismissed because it wasn't a government or university study. In Haryana, India, report after report shows that when buffalo are fed BT cotton oilseed cake, most have reproductive problems. Also skin problems, problems with their milk, and many deaths among the adults and the calves. I visited one village in Andhra Pradesh where they had allowed their buffalo to graze on cotton plants after harvest without a problem for 7 to 8 years. They allowed all 13 of their buffalo to feed on BT cotton plants for a single day, January 3rd, 2008, and within 3 or 4 days, all 13 buffalo had died. I asked them, how many of you personally have experienced itching from working in a BT cotton field? And it turns out most raised their hand. And they casually mentioned that they also lost 26 sheep and goats. Now, in India, Monsanto has purchased Mahiko Seed Company and has either convinced or forced farmers to plant this BT cotton. Now, all of the unpredicted changes that can occur on the level of the genome and the RNA, etc., can not only affect the health properties, but also the agronomic properties and the environmental properties. And so the cotton, it turns out, is not reliable. When it was planted in the United States for the first year, Monsanto had to pay out tens of millions of dollars in losses because the cotton failed. In India, there's a lot of problems with the cotton, so many farmers are unable to even repay the loans that they've taken out for the more expensive seeds and associated chemicals. The UK Daily Mail did a study to see how many of these farmers had committed suicide, unable to pay back their loans, and they estimated in 2008, 125,000. The estimate is now closer to 200,000. 12 cows died on a German farm after eating exclusively a variety of BT corn no longer on the market. About 24 farmers in the US claim that BT corn caused their cows or pigs to become sterile. In the Philippines, inhaled pollen may have caused disease, with all sorts of symptoms arriving only during the time of pollination, as well as unexplained human deaths and deaths among animals that were fed the corn. The same seeds were planted in 4 more villages the next year. The same symptoms returned during the time of pollination. Now, the third reason why we may see problems with GMOs is that the protein that is produced by the inserted gene may be different in the new organism. And this can occur in many ways. When you insert the transgene into a crop, it can be truncated, mutated. In fact, that MON810, which had that new allergen, 30% of the transgene was lopped off, truncated. And the protein that it produces is kind of a fusion protein between that which is dictated by the transgene and also the plant genome. Another one has a point mutation. Genes can be read differently. They can produce multiple proteins. And so the amino acid sequence of the protein may be different. But even if the amino acid sequence is the same as the original, it might be folded differently or have different glycosylation patterns, different molecules attached to it, which again can turn a harmless protein into a deadly one, which appears to be what has happened in Australia with some peas that fortunately never made it to market. Now, they took genes from kidney beans and put them into peas in order to create an insecticide. And the peas had passed all of the studies that are normally done before GM crops get put on the market. So they were ready to be introduced, grown in Australia, and shipped to India for consumption. But the developers decided to do one more study that had never been done before by any GM food crop developer and never done since. It was a mouse model with an immune test, 5 of them actually. First, they applied the protein in its natural state from the beans to the mice, no problem. Then they took the identical protein with the identical amino acids and applied it to the mice, and they had 5 inflammatory reactions, suggesting that the harmless protein has been changed somehow and could now create potentially anaphylactic shock. So they canceled the development of the peas and looked further to find that, in fact, there was a subtle change in the sugar molecules, the glycosylation patterns, which they then blamed on the change. When the Monsanto spokesperson in Europe was asked to comment on these developers' peas in Australia, he proudly said, "This shows that the regulatory system works." It actually shows how they spin the information 180 degrees, because the regulatory system never required the advanced mouse model. In fact, none of Monsanto's products had been submitted to the mouse model. And they don't move genes between closely related species. They move genes between kingdoms, from bacteria and viruses to plants. The fourth possible problem is that there's more herbicide residues on herbicide-tolerant crops. The plants essentially drink the poison and often store it in the soybeans, for example, in the part that we eat. Chickens fed a herbicide-tolerant Liberty Link corn died at twice the rate, but this was an industry-funded study designed so poorly that even a doubling of the death rate was not statistically significant. Now, in terms of herbicide, Roundup is definitely the one that is most used. And in a study of herbicide use in the United States, there was an increase in the first 13 years of 383 million pounds. And because Roundup is becoming less effective on these herbicide-tolerant weeds, farmers are using more and more of it. So in the last year of the study, the increase was 31%. Now, it's interesting about Roundup, and I think you'll appreciate the qualities in this conference more than other places. Roundup was not originally patented as an herbicide. Its original patent was as a broad-spectrum chelator, binding with a wide variety of trace minerals and other minerals, keeping them unavailable to the plants. And that's how it works. It reduces the plant's defenses against disease. Then it reduces the population of beneficial organisms, microorganisms in the soil, which normally provide nutrients into the roots and also keep the population of the disease-creating microorganisms down. And then also Roundup, or its active ingredient glyphosate, promotes the diseases in the soil, and they do the killing of the plants. Glyphosate is not directly toxic on a plant sufficient to kill it in sterile soil. What it does is it creates a perfect storm to weaken the plant and strengthen disease. And it lingers in the soil, affecting the nutrient profile and health of plants that are subsequently planted in that same field. Now, it's dramatic how much nutrients are taken out of our food supply. Here's an example of the reduction of nutrients in a plant for iron, manganese, and zinc, both a reduction of uptake and transport. Very dramatic. In 2007, 184 million pounds of glyphosate-based herbicide was sprayed. Consider all the nutrients that have been removed from the food supply as a result. Now, alfalfa nutrients are dramatically reduced when you spray it with Roundup, and the introduction of Roundup-ready alfalfa does not bode well for the health of the animals, particularly the dairy cows that will be fed the alfalfa hay. The primary ingredient for livestock food in the United States is Roundup-ready crops: soy, corn, cottonseed cake, canola mash, sugar beet pulps. All 5 Roundup-ready, and they just approved alfalfa. There is a chronic deficiency of nutrients in the livestock eating these products. I interview veterinarians who say there is a universal reduction in manganese, for example, which is one of the most serious of the nutrient deficiencies. One person sent a liver to a laboratory to test. No detectable manganese in the stillborn calf's liver. So we eat the animals who are sick and deficient. We eat the plants, and we in turn may not only be getting the nutrient deficiency that's passed on from the Roundup, but also we're getting traces of Roundup itself, which is linked to birth defects. It kills placental cells, human placental cells. It's linked to lower sperm counts, abnormal sperm, and cancer. In addition to creating disease to kill the plant, it's promoting diseases around the United States. Over 40 plant diseases have been linked to the increase in Roundup, including diseases linked to Fusarium, which in turn creates mycotoxins, which have been linked to serious outbreaks of disease throughout history. As you plant year after year in cropland that's sprayed with glyphosate, you can see the difference on the right versus the left of the quality of the crop after 10 years of applying glyphosate. There is a new organism that has been identified by a group of scientists, and there was a secret letter sent to Secretary Vilsack describing this new organism that looks like a fungus, but it's the size of a virus. And it appears to be in high concentrations of Roundup-ready meal and Roundup crops, and in crops sprayed with Roundup, and in lower concentrations in products that are not sprayed with Roundup. It seems to be in high concentrations on the leaves of plants that are diseased and the aborted fetal tissue of livestock that have had miscarriages. In fact, Koch's postulates has been applied, so it's been able to be introduced to animals in a laboratory, inducing a spontaneous abortion, and then re-extracted and cultured. So it appears to be linked with spontaneous abortions. So it is being evaluated. The reason I'm talking about it now, even though it's supposed to be secret, is that the letter that was sent to Secretary Vilsack was leaked onto the internet, and that's why we're talking about it. Normally, this type of information would be exposed to a lot more research before it was made public. The fifth possible problem is that genes inserted into GM crops might transfer to our gut bacteria or into our own DNA. The only human feeding study ever conducted, brilliant study, took 7 human volunteers. They had ileostomy bags. They had their lower intestines removed, not for the study. They fed them a soy burger and a soy milkshake and looked inside the ileostomy bag, and there, lo and behold, they were surprised to find how much intact genetically modified soy had survived passage through the stomach and small intestine. But in 3 of the 7 volunteers, before they were fed the meal, embedded in the DNA of their gut bacteria was the gene that had been found in the bacteria in the chemical waste dump near Monsanto's factory. In other words, they had eaten genetically modified soy in some previous time, and this was in the UK, where they don't eat a lot of GM soy. And the gene had jumped into the DNA of their gut bacteria. And it was in there on a stable basis with the promoter, which is the on switch, and they had Roundup-ready gut bacteria. Now, this may give a survival advantage to bacteria when we have so much Roundup in our diet that normally has antibacterial properties. So the Roundup in our diet may be stripping us of bacteria except that which has the Roundup-ready gene, so we may be colonizing the gut bacteria of North Americans. Now, if you eat a corn chip, and the corn chip has been made with BT corn, which is highly likely, and that gene would also transfer, and no one has studied to see if BT genes transfer, but if it does, it might turn our intestinal flora into living pesticide factories. And BT is known to break open the stomach of insects and kill them, and some people believe it may be linked to leaky gut. Now let's take a look at GM soy, just to see if there's evidence of problems. Now, soon after GM soy was introduced to the UK, the York Laboratory reported an increase of soy allergies by 50%. No follow-up was done to see if GM soy was the culprit. But there are many reasons why it might be. When GM soy was fed to mice, there was a dramatic reduction in enzymes produced in the pancreas. If it takes longer to digest protein, it gives longer time to produce an allergic response. One study that needs to be repeated with better controls showed a new allergen in GM soy. We already discussed the 7-fold increase in trypsin inhibitor, a known allergen in GM soy. Herbicide residues are increased. Even the Roundup-ready protein that's intended to be in the soy has properties of a dust mite allergen. And that is the one that's continuously produced inside our gut, possibly triggering allergic reactions on a continuous basis. Here's a picture of the liver of rats on the right that were fed GM soy compared to those on the left. Testicles changed from pink to blue in male rats fed GM soy. I'll let that one sink in. Mice fed GM soy had changes in their testicles, including damage to the young sperm cells. Female rats fed GM soy had changes in the uterus and ovaries and hormonal balance. Offspring of GM-fed mice had changes in the way the DNA functioned in the embryos. I gave a talk at the EU Parliament a few years ago with Irina Ermakova, a senior researcher of the Russian Academy of Sciences. She gave me these slides. They're Russian-speaking rats. They volunteered for a soy feeding experiment in which, starting 2 weeks before they got pregnant, they were fed GM soy, and it continued through pregnancy and lactation, and more than half of their babies died within 3 weeks, compared to a 10% death rate of the group who was fed non-GM soy. There was also a change in the size. Many of the GM-fed group were quite a bit smaller. And in a subsequent study, they were unable to reproduce. Now, as is typical of researchers who find adverse findings, Dr. Ermakova was attacked, victimized, and she told me her boss was under pressure from his boss, so she couldn't do any more GMO studies. One of her colleagues tried to actually comfort her by saying, well, maybe the GM soy will solve the overpopulation problem. One of the appropriate criticisms of her research was that she never did a biochemical analysis of the feed. So we don't know if there was some toxin in her particular bag of soy flour that she used that might have caused the astounding results. But after doing it 3 times with similar results, coincidentally, the entire facility switched to a GM soy-based rat chow. So she asked her colleagues after 2 months, what's your infant mortality rate? And it had skyrocketed to over 55%, suggesting it wasn't a particular toxin in her feed. In another Moscow experiment, also Russian Academy of Sciences, hamsters fed GM soy over 2 years. By the 3rd generation, most lost the ability to have babies. They grew at slower growth rate, 5-fold increase in infant mortality. Some had hair growing in their mouths. If GM crops are so bad, why don't we see more problems? Well, the L-tryptophan epidemic gives us a hint why not. It took 4 years to discover the epidemic, and yet it was screaming to be discovered. The symptoms had 3 simultaneous characteristics: new, acute, and fast-acting. All 3 had to be in place for them to identify the cause of the epidemic. What if it was not new symptoms? What if it were cancer or heart disease? No one would check to see if you're taking L-tryptophan. What if it were mild symptoms like frequent colds or memory loss? Or memory loss? Or what if it was like the hamsters where it took the 3rd generation to show the sterility? So what about the GM crops on the market? When they were introduced in 1996, 7% of Americans had 3 or more chronic illnesses. 9 years later, 13%. Food-related illnesses doubled between 1994 and 2001. Reproductive problems are on the rise. Gastrointestinal problems are on the rise. Autism, food allergies, inflammation, etc. We can't responsibly say that GMOs are the cause or a contributor, but we can't responsibly say they're not, because there's no post-marketing surveillance, no human clinical trials, no proper evaluation of all the changes that occur. Approvals are based on assumptions, many of which have been proven false, and on industry research, much of which is rigged to avoid finding problems. So I'd now like you to rate yourself from 1 to 100, how vigilant you'll be next week at avoiding genetically modified foods. So how many people will be low vigilance? 1 to 20. Raise your hand, 1 to 20. 20 to 40. 40 to 60. 60 to 80. 80 to 100. Some difference there.
How do you know?
Yes, how do you know? I have a non-GMO shopping guide for every one of you at the book table outside. And there's also packets of them if you want to give for your patients. How active do you wish to be? Now, before I talk about how active do you plan to be on educating patients on GMOs, I want to share with you, since one of the themes today is actually getting people out there and doing something to change the structure of our health. It turns out that getting rid of GMOs is not that difficult. Remember the potato study and Dr. Arpad Pusztai? Well, when his gag order was lifted, 750 articles were written within a single month, and 10 weeks later, the tipping point of consumer rejection was achieved. And within 1 week, virtually every major food company committed to stop using GM ingredients in Europe, but not here. We think as little as 5% of US shoppers avoiding GM brands could create a tipping point. Why? Because GMOs offer no consumer benefits. The same companies that are selling us GMOs here have already taken them out of Europe and Japan. They can replace GMOs without even changing their recipe, just going to the non-GM corn and soy. So we believe that by inspiring as little as 5%, and maybe we're off in our numbers a little bit, but it won't be much, we could end the genetic engineering of the food supply. So I add that as an extra motivation to participate, and I want to ask you now how active you would plan now to be in educating patients about how to avoid GMOs. So how many people plan to be particularly low? 1 to 20, raise your hand. 20 to 40, 40 to 60, 60 to 80, 80 to 100. So we just saw in 41 minutes how to end the genetic engineering of the food supply by telling people the truth. And it's not just doctors. I asked the same question to farmers, to food service directors, to parents, to lawyers, and they all, well, the lawyers have to be paid. Everyone changes their diet on the spot when they discover the problems. So our whole plan has 3 pillars of messaging. 1, the health dangers. 2, how to avoid GMOs. And you can also, here's the shopping guide you can get. You can also go to iPhone application Shop No GMO, or go to our website, nongmoshoppingguide.com. How to avoid GMOs and the fact that a tipping point can get rid of GMOs. So that's how we think we can end the genetic engineering of the food supply, and it's already happening with bovine growth hormone. We're part of a coalition that has been educating people about the health dangers. Bovine growth hormone has been condemned by the American Public Health Association, condemned by the American Nurses Association, banned in Canada, banned in Europe and Japan and Australia and New Zealand, and now it's banned in Walmart's milk, and in Dannon and Yoplait and Starbucks and most American dairies. And it never enjoyed the high-profile coverage that occurred in Europe. It was passed by healthcare practitioners and health-conscious shoppers and parents of young kids. So it demonstrates that a tipping point can occur in the United States associated with genetically modified products. And in fact, it's already happening. The Nielsen survey in 2009 said the fastest growing store brand claim was non-GMO. Year to date, it's the 3rd fastest overall health claim on packaging of products sold in grocery stores. In fact, Supermarket News said in 2009 that 2010 would show an unprecedented upsurge of consumer awareness and concern about GMOs, and that's what happened. I've been traveling around the United States for 8 years, speaking on the health dangers. I have never seen such an upswell of educated, concerned, and active consumers and physicians. I go to doctors' offices where they prescribe non-GMO diets to every patient. They give out our audio CDs to every patient. They have our texts. They have our videos. They play videos while people are getting IVs. Talk about a captive audience. The American Academy of Environmental Medicine, in May of 2009, urged all doctors to prescribe non-GMO diets to every patient, citing reproductive problems, immune system problems, accelerated aging, dysfunctional regulation of cholesterol and insulin, organ damage. I talked to doctors like John Boyles, who said, I used to test for soy allergies all the time, but now that it's genetically engineered, it's so dangerous, I tell my patients never to eat it unless it's organic. We believe that education is the key, so we have been strategically designing education to do the reach out so that individuals don't have to become experts. So we have the right brain book, the left brain book, the audio, the podcast, the series of articles, etc. But we're also teaching people to speak on GMOs. We've taught 450 people in the last 12 months to speak on GMOs in one-day workshops and 4-part webinars. And so we would like to invite you to help end the genetic engineering of the food supply simply by helping your patients understand the risks and make healthier non-GMO choices. So we have non-GMO health risks brochures, which you're able to take a free one at the booth, at the bookstore at the end of this lecture. And you can also sign up for our free electronic newsletter, Spilling the Beans, or leave your business card so we can tell you more information as it comes on. And for all the references in this talk, I refer you to Genetic Roulette, which is written as a 2-page spread. The left side is a summary for the attention deficit politician. The right side is the cited detailed text. So what I see now is an amazing opportunity. Because GMOs give no consumer benefits, because these companies have taken them out already, and because we now have sufficient evidence that simply by telling the truth, we can change people's diet on the spot, we can take back our food supply and together reclaim a non-GMO world. Thank you very much.